KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video, so we are going to discuss pollen tube in this style. Okay, means of pollen tube in the style means here after formation of a pollen tube, means after completion of pollination, pollen tube formation takes place. No, so that one after formation of a pollen tube in this style, what is happening and what are the types of styles are there? Everything. It is new information. So I have been gathered from the different types of materials. So listen carefully. New information only, but easy da. No worry. So now let us start to discuss pollen tube in this style. After the germination of a pollen tube enters into the style from the stigma, isn't it? Means when the particular pollen tube is germinated, so definitely what is a pollen tube is germinated on the stigma, no, definitely that will be enters into the style region. Okay, next one, the growth of the pollen tube. The growth of the pollen tube in the style depends on the type of the style. Okay, means once you see the growth of the pollen tube in this style depends on the type of this style, means here like a growth of the particular tube or pollen tube, definitely that will be differs from the one style to another style. Now here once you see there, no, the types of styles, three types are there. So there are three different types of styles are there. So there are first one is holotype or open type or open style, and second one is a solid style or closed style, and third one is a semi solid or off closed style. Okay, so there holo or open type, solid or closed type, third one is semi solid or off closed style. Okay, so first one is holo style or open style. Holo style point of view or open style point of view, what is the information? So we have to save in our mind, means it is a common, which one? Holo style or open style point of view, it is common among the monocotyledons, or it is a common among monocotyledons. So means in this particular holo style or open style, where we can find out, so very commonly we can watch in monocotyledons. Next one, a holo canal running from the stigma to the base of the style. So means is present, means once you see the holo canal means empty canal. Suppose if you take tubes are there, no, without water, how we can find out the empty space within the tube, no? So likewise a holo canal running from the stigma to till the base of style we can observe. Okay, so holo style or the open style of phenomenon or that kind of a situation we can watch from the stigma to the style. Okay, now then what is the main important point? No, the canal is filled with secretions. The canal is filled with secretions, and means here empty space is there now. So this particular what is the canal is there, no, that one filled with secretions, which serves as a nutrition for growing pollen tube. Means here definitely if you see there in a holo tube type of like a style, you know, we will be taking as a open style, and this one we can observe in monocotyledons, and a holo canal, so means here the empty canal is there, no, that will be situated from the stigma to the style. Okay, stigma to base of the style. Now this particular what is a canal or like a tube-like structure is, you know, that one filled with secretions which serves as nutrition. So means because within that canal, so that particular canal is filled with secretions. So these secretions only which serves as nutrition for growing pollen tube. So means while that particular pollen tube growing through that particular empty space or a hollow space, no, so that one that particular canal is filled with the secretions, no, so these secretions definitely helps. These secretions definitely helps for what? In growing of pollen tube. So why? Because these secretions serves as nourishment tissue for growing of pollen tube, and also controlling which one, and these secretions or these kind of chemicals, no, and also controlling incompatibility reactions. Okay, incompatibility reactions between the style and pollen tube. Means here these particular secretions also controls, these kind of secretions also controls which one? Compatibility reactions between the style and pollen tube. Next one, the secretions contain. So means if you see there, before we seen what is the point? This secretions serves as a nutrition point, or having of or service as a nutrition, means definitely those secretions must have some kind of what are the things, some kind of nutritive like elements. So what are those? Carbohydrates, lipids, and some like some enzymes also even present there. Means so carbohydrates are present, lipids are present, along with all these things, no, some enzymes. So some enzymes like esterases. Am I spelling you once you see there? ESTERASES, esterases. Okay, esterases, acid phosphates. Esterases and acid phosphates, as well as compatibility controlling proteins. Okay, esterases and acid phosphates and compatibility controlling proteins also are present. So in the hollow style or open style. Okay, the hollow style or the open style, so we can see very familiarly in the monocotyledons. So in this particular, what is the empty space is present from the stigma to the style, you know, so there that is completely filled with a few secretions. So these secretions are helps in nutrition point of view, and what is the important point? Next one, it also controls, means what are the secretions are present in the like hollow canal, no, also involves in serving of, like once you see there, also controlling the incompatibility reactions between the style and pollen tube. Okay, next one, the secretions contain carbohydrates, lipids, and some enzymes like means here nutrients are there. So because of their nutritive nature, no, so what are the chemicals or what are the things we can find out? Means carbohydrates, lipids we can find out, and some enzymes like esterases and acid phosphates. Esterases and acid phosphates we can observe, as well as compatibility controlling proteins also are present. Okay, so next one, solid style point of view. Second one is solid style or closed type of style. If you see there, no, this kind of style very common in dicotyledons. Means solid style or closed type, it is common among the dicots. It is common among the dicots. It is characterized by the presence of central core of elongated and highly specialized cells known as transmitting tissue. This is important. So means maximum times, no, here this particular style chance is to ask. So in this particular styles, no, what is the main important? So commonly we can observe in dicotyledons, and it is characterized by the presence of central core of elongated and highly specialized cells. So elongated highly specialized cells are present. These elongated highly specialized cells we will be taking as transmitting tissue. Don't forget. So means here the transmitting tissue presence we will be observing only in solid style. Name mattum solid, just there they have given a name as solid, but it is not being like very solid. Solid means general meaning we know very well, but our subject point of view, that meaning you don't amplify. Okay, solid means they have to give one name, that's why they have given as a solid. But why they have given as a solid means because of the presence of elongated and highly specialized cells known as transmitting tissue. Okay, so this particular transmitting tissue also, how this particular hollow canal consists of nutrients or the like secretions. Here transmitting tissue also contains secretions. Transmitting tissue also contains secretions like holostyle. Already we discussed this. Means how this particular holostyle consists of secretions. Likewise, even solid style. Solid style or closed type of style also consists of secretions. Okay, next one, the pollen tube grows through the intercellular spaces of the transmitting tissue. Means here one important point, one more point. What is the point? So here what is the pollen tube is present. Once you see how much clearly they have given. So what is a pollen tube is there, that particular pollen tube grows through the intercellular spaces of the transmitting tissue. So means if you see the structure of a transmitting tissue, it is made up of group of cells. So there between the group of cells most probably we can find out the presence of, we can see the presence of intercellular spaces. So here this pollen tube is enter through the intercellular spaces of the transmitting tissue. Okay, so through the intercellular spaces. Next one, if you see there, third one, that is semi-solid or half-closed type. Semi-solid or half-closed type. This is intermediate between the solid and open type. It is intermediate between solid and open type. Means here authors also they have somewhat doubts here. That's why here somewhat doubt information they have given, but that information is not required to you. But semi-solid or half-closed type means it is intermediate between the solid and open. If open type, if you see there, there empty space, the hollow space or hollow canal the irika. And that particular entire empty space is occupied by some kind of secretions that we have seen. Likewise, a solid style or the closed type, if you see there, there which one is there? Elongated, highly specialized cells are present. These elongated and highly specialized cells we will be calling as transmitting tissue. In a hollow space or open style, we are observing the presence of empty space or a hollow space. So how we have written now, like that only, let us learn hollow canal. Next one is solid style or the closed type, we are observing the presence of transmitting tissue. So likewise, next one, what is the difference here we can find out. So hollow canal consists of secretions, so which acts as a nutrition tissue or which contains of nutritions. So what are those? Carbohydrates and lipids. Likewise, even solid style or closed type also consists of specialized tissue. So this particular specialized tissue name is transmitting tissue. This particular transmitting tissue also contains secretions. Secretions are there means there also definitely we can guess the information that is the presence of carbohydrates, lipids and some enzymes. Now here the semi-solid point of view, it is like being like or it is like it is situated between the both. One is a semi as well as open type. Okay, next one, next concept point of view. So here what is the information here we can say means entry of the pollen tube into the ovule. If you take entry of the pollen tube into the ovule, so here what are the main important points to be discussed means entry of the pollen tube into the ovule. There are three types of pollen tube entry into the ovule. So there are definitely three different types are there of pollen tube entry. So there are three different types are there. So first one is porogamy, second one is chalazogamy, and third one is misogamy. Porogamy, chalazogamy, misogamy. One more time, try to say along with me. Porogamy, chalazogamy, misogamy. If porogamy point of view, so what is the meaning of porogamy? How this particular process occurs means, if suppose when the particular pollen tube enters through the micropylar region, if to where it is moving, so once you see the side heading, the entry of the pollen tube into the ovule. Emma, so now here the pollen tube is entering into the ovule. My dear students, see here one point you try to save in your mind. What is that? Here the pollen tube is entering into the ovule. Once the pollen tube is entered into the ovule, then only it can enter into the embryo sac. Why? Because embryo sac which is situated within the ovule now. Emma, within the ovule we can observe the presence of embryo sac. So thereby here definitely there we can find out three different types. So first one is porogamy, second one is chalosogamy, and third one is misogamy. Now porogamy point of view, when the pollen tube enters through the micropylar region, suppose if the pollen tube enters into the ovule through micropylar region, one point porogamy means pollen tube enters through the micropylar region. Next one chalosogamy. In a question only, answer also we can find out. In chalosogamy, what is the point? So if the suppose when the pollen tube enters through the chalosal region, so definitely when the pollen tube enters through the chalosal region, so that particular phenomenon we will be taking as a chalosogamy. Next one, misogamy point of view. Suppose if the pollen tube enters through the integuments, suppose if the pollen tube enters through the integuments, if the pollen tube enters through the integuments, so such kind of thing we will be taking as misogamy. So thereby here there are three different types of entry we can find out, entry of the pollen tube into the ovule. So three types we can find out. First one is porogamy, second one is chalosogamy, and third one is misogamy. Now here one more information. Sir, if the entry of a pollen tube into the ovule, three modes are there now. Then out of these three, so what is the mode generally pollen tube use for the entry of this particular pollen tube into the embryo sac? How sir pollen tube enters into the embryo sac means out of these three types. Once you see the information, you will understand. So the entry of a pollen tube into the embryo sac. What is information? No, irrespective of the place of the entry of the pollen tube. Once you see, irrespective of the place of the which one entry of a pollen tube into the ovule, it enters into the embryo sac at micropylar region. Means the modes, there are three different modes are there for the entry of a pollen tube into the ovule. Okay, porogamy we seen, chalosogamy we seen, and misogamy also we seen. But even though these are all different types are there, but entry of a pollen tube into the embryo sac takes place only through the micropylar region. Means so definitely, so here the pollen tube enters into the embryo sac only through the micropylar region. Next one, the pollen enters into the embryo sac directly into one of the synergids. So here definitely what is a pollen tube or pollen tube is there? No, that particular pollen tube enters into the embryo sac directly into the one of the synergids. So thereby here one point we have to get conclusion. What is that? So here the two synergids are present. So what is a pollen tube is entering into the embryo sac, that one enters into the embryo sac through synergids. Okay, through synergids. So thereby while entry of a pollen tube into the embryo sac, one of the two synergids are ruptured. Okay, one of the two synergids get ruptured. Next one, the growth of a pollen tube, the growth of the pollen tube towards the ovary, ovule and embryo sac is due to the presence of chemotropic substances. Once you see there, here once you see the growth of the pollen tube, that is growing towards the where? Towards the ovary and towards the ovule and towards the embryo sac. Ovary we see now. So there likewise it is moving through these kind of parts due to the presence of what? Due to the presence of chemotropic substances. So there some kind of chemicals. So because of the presence of chemotropic, because of the presence of chemical or because of the presence of some kind of substances, so here this particular pollen tube enters like this through the ovary and through the ovule and through the embryo sac. Next one, the pollen tube. The pollen tube after the traveling. So the pollen tube after the traveling, the whole length of the style enters into the ovary locule. Okay, once you see one more time, when the pollen tube after traveling the whole length of the style enters into the ovary locule. First of all, what is a pollen tube is there? No, that will be enters into the ovary locule. Ovary locule, where it is guided towards the micropylar region or the micropyle of the ovule. So by the structure known as obturator. So here the obturator is a special special structure is there. So what is the use of this special structure means? So once you see where it is guided towards the micropylar region, means it is the pollen tube. First of all, it is enters into the ovary locule. Ovary locule means empty space within the empty space within the carpel, we can be taken as locule. Okay, so now here from here empty space to that particular pollen tube moving towards the micropylar region. So by a specialized structure known as obturator. So this particular point also is important. Next one, here suppose sir, what is the importance of or difference between the obturator and which one filiform apparatus, finger like structures means here the filiform apparatus is belongs to the, actually in our textbook they mentioned, so the specialized structures which are belongs to the belongs to the synergids having finger like projections and helps to that or helps to the sending off or guides the entry of a pollen tube into the embryo sac we see now. So likewise here also obturators, the obturators helps for what? When the pollen tube traveling the whole length of a style enters into the ovule, means now here the pollen tube is there in a ovary locule. So from here to, so that particular pollen tube is moving towards the micropylar region. Okay, so means where it is guided towards the micropylar region of the ovule by a specialized structure known as obturator. Once the particular pollen tube reaches to the micropylar region, at the micropylar region again because of the presence of a filiform apparatus, that particular pollen tube enters into the embryo sac. So that particular information so to be studied very neatly. So next information is double fertilization and triple fusion. That is double fertilization and one more is triple fusion. So now double fertilization and triple fusion, so this information as biologists or as biology students we know very well and we have studied. So now the double fertilization and triple fusion point of view, SG Navanshin or surnames are not required, Navanshin and once you see GUIGENARD, gonad means here Navanshin, only you try to remember. So this particular scientist name I have taken from the our metric textbook. So Navanshin and gonad, so observed in lilium as well as a fritillaria. Okay, so Navanshin and gonad observed which one? Double fertilization and triple fusion where in lilium and fritillaria. That what they were observed means that both the male gametes released from male gametophyte. Male gametophyte. Male gametophyte means pollen grain only when the particular generative cell divides into two male gametes, we can be taken as male gamete or male gametophyte. Okay, now these two male gametes, both male gametes we have written like this. So both male gametes are released from the a male gametophyte are involved in the fertilization. Okay, so are involved in the fertilization. Since both the male gametes are involved in the fertilization, so this phenomenon is called as a double fertilization and it is unique to the angiosperms. So means here the process of double fertilization or here the process of fertilization or the double fertilization only let us take. Okay, why? Because here their double fertilization is nothing but the combination of two different types of processes. One is syngamy and one more is triple fusion. So syngamy and triple fusion both together we will be calling as double fertilization. Okay, both we will be calling as double fertilization. Double fertilization so contains two processes combinedly. One is Misogami and one more is triple fusion. Both together we will be calling as double fertilization. So here first of all let us start to discuss about the fertilization. Don't forget, so this particular double fertilization process is unique in angiosperms. Don't forget, is a neat point of view important. Suppose if you take a double fertilization, what is a double fertilization? Or right here the double fertilization only we can see in angiosperms, and other species we have seen there are different species or the different types of species are there, but all these members are not showing double fertilization process, only we can see in angiosperms. Next information is now Misogami means Misogami or the fertilization. Misogami means when out of two male gametes, one particular male gamete fused with egg cell, so results in the formation of diploid zygote. So results in the formation of diploid zygote. That particular diploid zygote is a connective link. Okay, so that particular diploid zygote is acting as junction, or means here we will observe one point. No, so what is the point we will observe here? If you take any particular, so suppose if you take any particular thing, so here definitely the link of generations, so definitely starts with the link of generations, so definitely starts from the zygote. That's why this particular zygote we will be taking as a connection link between the generation to generation. A point you understood. So thereby, so here this particular diploid zygote, so this particular diploid zygote later after somewhat divisions, so converted into embryo. Later triple fusion point of view. Triple fusion point of view, what are the main important points to be discussed means? So what are the main important points to be discussed? No, triple fusion is nothing but when male gamete fused with two polar nucleus. When male gamete fused with second male gamete, when second male gamete fused with polar nucleus, so there it results in the formation of triploid primary endosperm nucleus. So that particular triploid primary endosperm nucleus later develops into endosperm. So that particular endosperm later acts as a nourishment tissue, so which supplies the food materials to the developing embryo. So likewise here the process of double fertilization we can see. Double fertilization and the triple fusion. So first of all observed by and gonad, and in where, so lilium, and one more is a fritillaria. So this particular information we know very well. So here one new point we have learned, obturator. Next one, so here there are three different types of entries of the pollen tube we can see into the ovule. Likewise there are different types of types of styles also we have seen. So hollow style and solid style and semi-solid. Hollow style also known as open type, solid style also known as closed type, semi-solid also known as which one off closed type. So this is a small information regarding types of styles and entry of the pollen tube into the embryo sac. Thank you.